This video is a very general introduction so that you can get an idea of what to expect in the upcoming videos in this series. We've already talked about the information flow in a cell, which starts from the DNA, which is converted into RNA, and that information is then converted into protein. And this happens in the process of transcription and translation. I have an entire series dedicated to these two specifically, and if you're interested, go check that out. I have links to the playlist down in the description. The concept of DNA replication is not related to the conversion of information, but rather in maintenance of the information. And this is where the process of DNA replication comes into play. The maintenance of information is inherently linked to the cell cycle, which you may have heard as mitosis, meiosis, or fission in case of bacteria, or some other name. And the fact that we're talking about cell cycle suggests that there is some connection to the overall cell biology of a cell. For the sake of the depth in our understanding of DNA replication, this series will primarily explore the molecular biology side of DNA replication. There are some obvious questions that should come to your mind when you think about DNA replication. The first is the need. Why maintain information at all? The answer is not surprising at all because DNA has the information to keep the cells functioning, and you also need to maintain information if you want to pass it on to the progenies. And likewise, there are many reasons. The other simple question is, how does it happen? Well, that is what this series will be all about, so stay tuned for future videos. But let's get conceptual here. How does a cell know that it's time to start DNA replication? And is that time fixed? And if so, how does the cell know when to stop replication? The fact that there is a start and a stop suggests that there is something which has to do with the timing of the cell cycle. And this means that there must be an internal molecular clock in the cell to regulate this timing mechanism. There are many things, but let's formulate the problem of DNA replication more explicitly. We take the example of human cells. We have about 300 million bases, or 3 gigabases, or 3 billion bases in our cells. This translates to 3 times 10 to the 9 bases in total. Since we're diploid, there are actually twice as many bases to replicate. Now, the S phase of the cell cycle is typically around 8 hours in length. And that is fixed amount of time given to replicate 6 billion bases. 8 hour means that you need to copy or replicate 200,000 bases per minute. But the enzyme that copies the DNA, called DNA polymerase, can only copy up to 100 bases per second, which translates to a max speed of 6,000 bases per minute. But we need to copy 200,000 bases per minute. So how is this accomplished? The problem in humans, or eukaryotes in general, is also complicated by the fact that DNA is chromatinized. It is wrapped around histones. So DNA replication, as we will see later, is also coupled with histone mark replication. To quickly give you an idea, imagine that this piece of chromatin has a specific acetylation mark at a very defined location. So when it is copied, the histones are redistributed into the daughter DNA. And then you also need to reinstall the acetylation at the histone at the same spot as it was in the parent. So you're not just copying DNA, you're also copying its epigenetic profile. In this sense, you can think of DNA replication as chromatin replication. Here's another thing. Since DNA polymerase moves on the DNA to replicate it, do you think histones would block the movement of polymerase? We have seen the same sort of problem when we discussed the RNA polymerase movement. Anyways, with this series, I will attempt to paint a more realistic picture of DNA replication, something that is not very often appreciated. So how are we going to do this? As I said previously, we will focus primarily on the molecular biology parts in a fairly advanced level, and we'll start with the initiation step of replication, diving into the origin of replication, and various factors that are involved in this step, like helicase, primase, DNA polymerase, and many more. And in this, we will also look into different species, both in prokaryotes and eukaryotes. After that, we'll continue talking about polymerase and helicase complexes into elongation, as we unwrap some key terminology of leading and lagging strands. And we'll talk about different types of polymerases. The concept of lagging and leading strand will tell us how continuous and discontinuous modes of replication come together to copy the DNA. And this will hopefully lead into some of the classic pictures of replication forks that you typically find in textbooks as well. When looking at different polymerases, we'll compare the prokaryotes and eukaryotes, and we will see that there are many types of polymerases, and each of these types of polymerases have a specific function. And then we'll end with determination, and finally connect this entire process in eukaryotes specifically with histone mark replication, and the space and timing of the DNA replication in a cell.